Okay, so first of all, I want to thank the organizers for this very kind invitation. So, as they stated in their kind letter, the idea is to open discussions and open for collaboration. So, I will give you an overview of the things we have done in the past and end up with our most current research. So, the last slides are not published, so please refrain from pictures and so on. So, I start to have the time, because I've been told that I have less time than the others because it's too interesting. I mean, just told me. So I work at IBGC, which is an institute that dedicates a lot of energy to the study of mitochondria. And let's start with an overview. I mean, we all talk about mitochondrial dynamics and morphology, but this is something what I want to emphasize that is not new. I mean, we know that mitochondria are very, very diverse, and these are images from Pallade in this very old book, where we can see the different space occupied by the crystal or by the matrix. So we must be aware that variability and diversity is there from the very beginning of mitochondrial research. It's not something we have done in the 2000s with GFPs. It's something that was there. I have another example. Then the idea that mitochondria are filamentous also is something that it is made substantial to the appearance of photonic microscopy and GFP, but it was already there. Sorry. So it works. I didn't know. So here, for example, this image is very, okay, it's an electron microscopy image in a thick section by Pierre Favard, and it shows clearly the enrichment of the mitochondria and the epithelial side, where the energy consumption is taking over. These are images with rotamine 1, 2, 3 by L.B. Chen, which is a pioneer in the field. So obviously, dynamics is something very clear. And if I want to do more history, that was a surprise. I found myself in a seminar. This is a citation of a science paper in 1914, where mitochondria are almost never addressed, but are continually changing their position and also their shape. The changes in shape are truly remarkable, not only in the great variety of forms, but also in the rapidity. So this was almost not known to the public when we started to do the research with GFPs in the 2000s, and some people here in the audience were with me when we started to do this kind of stuff. So here you see a HeLa cell transfected with a GFP. Here you see this mitochondria that is changing side. So these mitochondria are all the time fusing and dividing. And since this was discovered, there are two main questions in the field. One is, how does it work? And the most important one for this audience is, for what it is good for or bad for. And so this is how we symbolize the whole thing. So mitochondria divide and fuse and divide and fuse. I mean, if we want to know who is better, the long or the short one, we will never find out. I always like to use, it's like a boxer, you know. A boxer is all the time jumping. You will only see the advantage of jumping when somebody hits. So I think mitochondria prefer to change shape between fragmented and elongated, because then they can react to different situations, okay? So imagine this is a boxer jumping and somebody hits. It will be better to move than to stay. So the main molecules have been identified, and this is not our work, it's the work of a lot of people. It's rather the mitofusins 1 and 2, OPA1, these are fusion proteins, and the fission proteins, DRP, and the advantageous factors. And it's the shift between both two reactions that determines the overall morphology, either elongated or fragmented. And we know now that this futile cycle of fusion and fission is essential for mitochondrial and mitochondrial DNA maintenance, for protein import and biogenesis, what determines the distribution of these molecules that are imported. And we need fragmentation and we need fusion for degradation by autophagy and mitophagy. This is not something we're specialized to. And there is a difference between doing movies and between determining an effect. So if you want to determine that mitochondria have fused, you better demonstrate that they exchange their contents, okay? We use a technique that's called PEG-mediated cell fusion, where we fuse cells with differently labeled mitochondria, and so we can demonstrate that indeed these mitochondria are doubly labeled, become doubly labeled, okay, and have fused. And we know that this is an active process because we can find areas where mitochondria remain singly labeled. Okay, here this green mitochondria, okay, it's not labeled in red, so it's an event. And what we are now exploring in the lab, and there are a lot of people, is what is the link between these dynamics and the bioenergetics. And in the one direction is how do bioenergetics modulate dynamics. We showed a very long time ago that it's enough to depolarize mitochondria with an ionophore or a secret molecule from the pharma industry. And then maybe the mitochondria do not fuse anymore, as is the case here, and they remain singly labeled. Not only singly labeled, but fragmented. So fusion-fission dynamics and overall morphology are modulated by bioenergetics, and this is also seen in cases where not such a drastic effect is imposed by, but rather, for example, Oxford's defects like in mitochondrial diseases. And Rodrigue has done a lot of work in this direction. So this is what happens, for example, when you add CCCP, that's how you achieve this massive microntation. So how are dynamics modulated by bioenergetics? This is an open question in the field, and I could not resist to show to this audience data that I always show to students when I do this lecture, and you will understand why I'm so excited. It's because we think about molecules that are most of them sitting in the cytosol or on the membrane and are GTPase and so on and so forth, and we forget what Hackenbron showed in the 60s. These are mitochondria isolated from rat liver, I think. They have this condensed conformation, and what he showed is that you simply need to add substrate to completely change the aspect. Then you can add a phosphate acceptor, and suddenly they become condensed again. You can add an inhibitor, and then they become more condensed, bypass the inhibitor, and they become orthodox. So if we can observe these dynamics at the ultrastructural level, what I want to emphasize is that it is very easy to understand that it can happen also at a more larger level. Okay? So mitochondrial ultrastructure is dynamic and is modulated by OXPHOS. So I understand that it's very easy that mitochondrial fusion-fission dynamics and morphology are also modulated by OXPHOS. And the other question that now we are addressing with Arnaud is how do fusion-fission dynamics modulate the OXPHOS? So in the other direction. Okay? So we are doing experiments in different directions. So this is the work of Celia Fernandez-Sanz in the lab. We knock out DRP1, and they do not work like I do with cells in culture, which is a very bad bioenergetic model. Okay? They work with a real tissue, which is the heart, which consumes a lot of oxygen and beats from the birth to the end. And they can study either the heart like this or after isolation of cardiomyocytes. And for example, here what Celia is looking is at this structure here, which is the mitochondrial associated membranes at the interface between the cerebral reticulum and mitochondria. And she is looking at the localization of DRP1, which appears to be very specific, okay, and seems to be regulated by modulators of mitochondrial and cardiac function. On the other hand, we are looking at the role of mitofusin. So this is a symbol of mitofusin, mitofusin 1 with the membrane topology, its conservation within all eukaryotes, even down to fungi, the presence of two homologs with redundant functions, mysterious functions in humans. And we know that these mitofusins are essential for mitochondrial fusion, required on both membranes. The function and molecular mechanism is rather unknown, and we know that mitofusin 2 mutations are associated to hereditary neuropathy. Okay. So we don't know, however, what are the consequences of these mutations of mitofusin functions. We don't even know how mitofusin functions. Okay. We don't know what are the physiopathological mechanisms of this disease, and the question is double, so you can read it again. Sorry. So to understand this, we first thought it may be interesting to see what are the bioenergetic consequences of a mitofusin 2 or mitofusin 1 knockout on bioenergetics, and to study this in a real tissue, not rather than in cells. Okay, and this is the work that Arnaud published before he joined IVGC and CNRS in France. So This is the knockout of mitofuscin 1 or mitofuscin 2. And in this case, he isolated cardiomyocytes, okay, from different backgrounds. And the first thing you see is that despite the fact that in all these movies, mitochondria can fuse all the time, mitochondria in cardiomyocytes that are very proximal, they are not fused to each other. Otherwise, you could not have mitochondria that are depolarized beside depolarized because they lack the TMRM fluorophore, okay, which depends on the membrane potential, and have the YFP. So you can have side by side mitochondria that are depolarized and polarized, so they are not fused all the time. Okay, and then what he could do is to show that the frequency of these depolarized mitochondria is much higher in mitofuscin 2 knockout cells and much lower in mitofuscin 1 knockout cells. And the difference between mitofuscin 1 and mitofuscin 2, two very similar proteins, remains still rather unknown. Okay, so he went further. I only saw a summary and showed that in the absence of mitofuscin 2, you lack a lot of coenzyme Q, and this results in very severe defects of enzymatic activities of the respiratory chain that may be complemented by coenzyme Q addition. So in a way, mitofuscin 2 diseases provoked by mitofuscin 2 mutations could be due to the bioenergetic consequences it induces. Okay? So now let's go back to other systems. So my personal question we are following now. So this is the case of Charcot-Marie-Tooth diseases, which are hereditary, motor, and sensory neuropathies. They affect mainly the peripheral nerves. Okay, 20 to 30% of one of these types, type 2A, is associated to dominant mutations of the mitofuscin 2 gene. Okay, and the work I showed you now was initiated by Thomas Rival, who is a researcher on Drosophila in Marseille, and asked me for advice and collaboration. So this is the sequence of mitofuscins, and here is a list of some of the mutations that have been found, essentially all over the molecule. And Thomas, he decided to focus on this one, R94Q and R364Q, which are the most abundant mutations, okay, observed in a large number of patients. And we choose two other mutations, I would say for control, but you will see it's not just for control. Let's see, I think 76, very near to this one, and T105M, which is at the GTP binding motif 1. Okay. So what Thomas did was introduce these mutants into Drosophila and express them in fly neurons. Okay? I insist to the fact that this is a dominant dominant disease. Okay, dominant. So here you see an alignment of mitofuscin and the homolog MAR. The mutations we have chosen are conserved. This is very advantageous for this assay. This is the expression system he uses. So essentially, all these flies, they only differ by a single nucleotide or a single amino acid, because we also introduce the wild-type MAR as a control. And this is just to show you that this is the expression of the endogenous and of the exogenous. So it's a situation very similar to that found in patients where we have one mutant and one wild-type phenotype gene, allele, sorry. And this is just to show you with a locomotor index, which is very easy to perform in flies, much easier than in mice. Okay, either they can fly or they cannot fly. So we see that all flies carrying mutations have a lower locomotor index. So we have a disease model, and now we want to know how it works. So the system allows to label specifically the mitochondria of the larval CNS. So here are the mitochondria. Okay, this is now enlarged. I have to move because I have. No, sorry. Okay. Okay. So these are the mitochondria in neurons in the wild-type situation. This is when we overexpress the wild-type MARF or mitofuscin, so a little bit more elongated. This is when the MARF is downregulated, they become more fragmented. And this is when we express very high amounts of mitofuscin. So we start to get this kind of globules. This is different from what we observe in mammalian cells. And these are the four different mutants. So what you see is a big clustering of R94 and T105. Okay, they cluster their mitochondria. They lose this normal morphology. And in the case of these two mutations, we start to see what we also see with an excess of mitofuscin, okay, this start globus. So we decided, because this somehow suggested that mitochondrial fusion may be enhanced, we decided to investigate whether these mutants are active. And for this, we went back to MEFs. So these are cells that lack both mitofuscins. Their mitochondria are fragmented. Okay, and we introduce wild-type mitofuscin 2, mitochondria picans filamentos. And when we introduce the different mutants. And what we see is that all these mutants can cluster mitochondria, these two ones, but the other ones can render them filamentous again. So they are fusion competent, essentially. Sorry. Okay. These two mutants are fusion competent. It somehow suggests that it may be not dominant negative, as one would expect, but dominant active molecules that would interfere with normal mitochondrial function through excess fusion. And the rest of the work is devoted to confirm this. So this is the first slide I showed you. I don't want to visit it again. And now what Thomas decided to do was to introduce DRP1, a fission molecule, in this genetic background. If this phenotype, locomotor phenotype, is provoked by an excess of fusion, we may compensate it by an excess of fission. And this is indeed the case. So you see, mobility is recovered in those both mutants. This is the slide I showed you before, okay, with this clustered mitochondria in the case of fusion of dominant negative molecules and the active molecules. And now, if he introduces DRP1, there is little effect on this ER3994Q, but here we restore normal mitochondrial morphology. So indeed, it turns out that the disease by this mutation is provoked by an excess of fusion. So he looked at several parameters. I show you here the last one. And this parameter is the distribution of mitochondria in neuromuscular junctions. Okay, mitochondria in green, junctions in red. And you see that all these junctions, or almost all of them, contain mitochondria. And that the number of junctions without mitochondria increases significantly if you express mutations, either active or inactive mutations. And that you can compensate by co-expressing DRP1. Okay, this is the numbering of that. So this is, you have the number of junctions with mitochondria is very low in the phenotype, and you co-express DRP1, it recovers. So how does it work? This is now the whole challenge. Okay, how do we get a dominant active molecule? So this, we were lucky enough, just this is in 2016, 1 year ago, and a few months later, the structure of mitofuscin 1 fragment was published, and this allowed us to show, to infer where these different mutations were. Okay? It turns out that both of the active mutations are in these 4 helix bundles, and that the dominant negative mutations are either in the middle of the GTP binding domain, this we knew, okay, or at the edge between the 4 helix bundle and this GTP binding domain. So this is a bacterial dynamic-like protein that resembles, and it's in a folded conformation. And so, well, we can speculate. I call this speculation. We can speculate that they somehow interfere with this domain, and so this may be altered, okay? Because the molecule is known to exist in 2 conformations, so here they may play a role. So that's where we are, I think. This is speculation. And so at the end, I would like to thank the people that did the work. So I'm working with Arnaud in IBGC. The people in the lab now is Arnaud, myself, Celia, here in the audience, Thibault, also Claudine and Elodie. A lot of work was done and initiated by Arnaud while he was still in the Max Planck, and he's still working with Eduardo. All the work with lights was done by Thomas Rival and Naija El Fissi at IBDM in Marseille. The DRP1 work in heart was initiated and is continued in collaboration with Professor Sheng-Shi Xu at Jefferson University in Philadelphia. And we want to go further, I mean, to do work with neurons, mammalian neurons, and this we'll do in collaboration with Pascal Berenguer and Christine Miquel, which are on Toulouse. And these are the organisms that finance our work, not only ANR, Initiatives d'Excellence, and IFM. Thank you very much for your attention.
I have a question, Manuel. Do you know, because what is the pathophysiological mechanism of hyperfusion? People think that usually fusion is more active for respiration. Is this mutant also inactive for respiration?
It's difficult to do biochemistry in flies, okay, but he has measured ATP levels, okay, not even ATP-ADP ratio, and ROS content, and so there are alterations there. But these are nerve tissue samples. I mean, it's not even neurons, okay? So it's something to explore this. We need neurons with fluorophore indicators or more precise dissections. But there are some hints in this direction. I really think that the image of fusing mitochondria doing well and fragmented mitochondria doing less well is not accurate. I really think that you need both, and if you hamper the normal cycle like we are showing here, then problems start.
I had another question related, but do you know whether this mutation changed the stability of the protein, and in the end, so in the hyperactive MFN mutation, you have more protein in the end, or the level is the same?
No, the level is the same. The level is not. I don't know in patients. Sorry, I don't know whether in patients all the patients have been checked. So whether the protein level is altered, but in expression systems, the protein behaves like the wild type.
Hi, thank you very much for your talk. You showed that when you use a depolarizer, you inhibit the fusion-fission cycle. Do you know what happens when you remove oxygen or when you cool down the cells? I mean, I'm preaching from our inquiry, obviously.
Yeah. There are a lot of studies done in this direction. So I've chosen the most dramatic one with CCCP or FCCP because it's obvious, it fragments. A lot of people, Rodrigo also, have done different drugs for different respiratory complexes, anoxy, and in general, everything that harms mitochondria tends to fragment mitochondria. But there are some discrepancies in the field or controversies because some cell lines are more sensitive than others. So everybody agrees that depolarization inhibits fusion and fragments mitochondria. As for the other treatments, there seem to be, it's much more difficult to find a consensus in the field.
Thank you.
More questions? So thank you, Manuel.